Thank you, Alex, and thank you all for coming and braving this potential apocalyptic situation we have going on here. So today I'm really excited to talk about a project that I've been working on since I started my postdoc here in August with numerous collaborators looking at kind of soil microbial communities in restored woodlands at Shaw Nature Reserve and kind of seeing how those microbial communities might impact restoration. And so we all know that human activities and agriculture and different commercial and residential development, as well as kind of industrial ventures, have really led to degradation of habitable landscapes. And we can see over the last 10,000 years, there's been a drastic decrease in numerous landscape types, particularly when we look at forests. There's been about a 1/3 decrease of habitable forests all across the globe. And this really can lead to altered abiotic conditions, different episodes of water runoff and temperature and CO2 can all change due to land degradation, as well as shifts in both above and below ground species composition, which eventually lead to changes in ecosystem function. And a response to this degradation is restoration ecology, in which scientists and restoration ecologists perform restoration activities for the goal of kind of improving native habitats, protecting native habitats, and eventually leading to kind of habitat recovery. And these activities can be anything from different management practices, potentially protecting native landscapes, reintroduction of native species, all with the goal, again, of protecting and recovering native landscapes. But the success of these restoration endeavors is highly variable. And so it's highly variable because they really often can overlook important below-ground factors. And ecosystem and ecological restoration practices have really traditionally focused on above-ground processes and above-ground plant communities. But the role of soil biota are really key for mediating plant responses to restoration, as well as plant-soil feedbacks can influence plant-to-plant relationships. So all of this can influence restoration success over long term. And soil microbes cannot be overstated in their importance. So they come in all shapes and sizes. We have a little bit larger with arthropods, and then of course we have protists in the soil, but really the main focus is fungi and bacteria, because those are the ones that we know the most about in their role in soil. And so fungi and bacteria really aid in nutrient cycling, as well as promote plant growth. And they even are important for pathogenic responses, which can mediate plant coexistence. Soil bacteria particularly are important for nutrient cycling and decomposition of very important things like nitrogen, carbon, and of course fungi fill obligate roles from the decomposer to pathogens, as well as very important symbiotic mycorrhizae. And the kind of advancing of research and our understanding of the soil microbiome and its role on these plant-soil feedbacks and restoration in general is really one of the key strategies laid out in order to kind of meet the UN Decade of Ecosystem Restoration goals. So it's really at the forefront of everyone's mind to kind of understand the relationship between soil microbes, restoration, and back and forth. But unfortunately, restoration management practices can also impact the soil microbial community composition. So things like reintroductions, soil inoculation, where you're adding either commercial or remnant soil to a site, thinning of invasives or even native plants, and prescribed burning can all have implications and cause shifts in the soil microbial communities. And so when we think about reintroductions, a lot of times we're considering rare and declining species, which are often remnant specialists. And what exactly is a remnant specialist species? It's a species of high conservatism value, over 7, and these high C-scores really indicate it has less tolerance for a degraded habitat. So they often fail to recolonize restored sites naturally, and they need reintroductions. But there's a big question about when should these species be reintroduced. So there's kind of a double-edged sword. So there's a thought that abiotic and biotic conditions are considered more favorable in later restoration stages. Maybe degraded sites have a missing symbiont in the soil that is found in later successional sites. But on the other hand, establishment barriers could occur in later restoration sites where there's priority effects taking place. Certain species that got there sooner can outcompete species that you were trying to reintroduce. So there's really this kind of dynamic of, is too soon, is too late, and trying to understand the relationship for these reintroductions of these high conservation species. Something that can aid in these reintroductions is soil inoculation. So soil inoculations are becoming increasingly popular in order to kind of facilitate successful plant reintroductions. But the results of these plant establishments with soil inocula are still variable. So they can be relatively positive. Adding mycorrhizae to the soil has increased plant richness in restored sites, but it can also be neutral. So forb growth has been shown to have really little difference between both inoculated and control soil, meaning the symbionts or the other microbes that they were looking to add to the soil were likely already there, or negative, in which commercial AMF, particularly adding this kind of commercial strain of fungi, had negative effects on biomass in dryland species. So again, it's very variable in how well these particular practices actually aid in reintroduction success. And of course, adding microbes to the soil will alter the soil microbiota. Thinning of vegetation is something else that often occurs in restoration ecology, in which you can thin native plants. So here, even in a managed pine forest, thinning out some of the understory trees and smaller trees led to an increased changes in beta diversity of the microbial community, but also led to a decrease in really important plant growth promoting bacteria, proteobacteria. And removal invasives often aids in native plant growth, which is really great, but it does shift the soil microbial community, which could have long-term effects on restoration as a whole. And of course, fire has major impacts both directly and indirectly on the soil microbial community. So prescribed burns lead to direct selection against heat-sensitive microbes. So almost immediately as a burn goes through, those heat-sensitive microbes are going to be diminished in the habitat. And also indirectly, fire can impact soil microbial communities. So it alters soil abiotic properties. Different nutrient levels will lead to the growth or abundance of particular microbes. And also it reduces the abundance of fire-sensitive host plants. So for example, AM-associating trees, like maples, are very fire-sensitive. They cultivate AM, but if they're removed in fire, it will have impacts on the soil microbial community. And microbes seem to respond variably to fire in general. Bacteria seem to respond neutrally or possibly positively, but soil fungi communities respond variably depending on guild, where pathogens actually respond relatively neutrally or positively to fire, while symbionts respond very negatively. So again, all of this is impacting the overall soil microbial community. And Shaw Nature Reserve allows us to kind of test some of these things and the response of the soil microbial community. It's a beautiful property. I'm sure most of us have been there. It's very large with multiple different habitat types, but they're also implementing multiple management practices that we can kind of look at and see how they're impacting the soil microbes. So they have prescribed burning, selective thinning, and of course plant reintroductions, which are key for kind of promoting native plants. And within Shaw Nature Reserve are 3 restored woodlands with highly similar vegetative and soil properties, but they vary in their restoration age, which creates what's called a chronosequence of sites. So you have a younger restored site that's only experienced 1 or 2 burns. It's only been in a restoration phase for a handful of years. You have kind of an intermediate woodland that's experienced more burns, but still not as much so as our oldest restoration site, which has experienced numerous burns over many decades. So it sets up this really nice gradient of restoration so we can test some of these things. And again, we're really trying to look at the above ground, below ground restoration kind of theme here and its influence on soil microbial communities. It's a multi-layered feedback dance you could think of between soils, plants, restoration, and back and forth. So the overall goal of this particular study was to understand how soil microbial communities vary within a restoration age chronosequence and how different remnant woodland specialists might perform when planted in those plants, in those soils, pardon me. And so we tested the growth responses of 4 herbaceous plant species to these whole soil inocula treatments from the restoration sites. used DNA metabarcoding and amplicon sequencing to really quantify the microbes that were in the soil. So this is an outline of an extremely detailed project that was multi-phased, multifaceted, but I really want to get into this so that everyone kind of understands exactly what we did. So we first characterized the soil and vegetation properties of these 3 sites that I mentioned. So we had our young restored site. It's only been in restoration phase for 7 years and it's experienced 2 burns. Intermediate has been in restoration phase for 18 years with 6 fires. And our eldest site, 29 years of restoration with 7 fires. And again, we characterized both soil properties and vegetative properties. Then we collected the soil and we did the special metabarcoding to see which species were in the soil. We also used that soil to do a whole inocula experiment where we planted 4 herbaceous species in each soil type and we looked at their growth and development. Then, because plants influence the soil and soil influences the plants, we then metabarcoded this conditioned soil to see how does the conditioned soil vary from the original inocula soil, which could have implications for long-term restoration success. If you're adding plants, it's going to change the microbial community. So our first objective, again, was to characterize these sites, understand possibly where they may be different in terms of what our eyes can see, vegetative and soil properties. And we expected differences in these properties because they are different woodlands. But we also, of course, expected differences in the soil microbial communities because they're different sites with different management histories, which we know can lead to differences in the soil microbes. And of course, prescribed burning reduces the abundance of symbiotics and fire-intolerant species and can promote the abundance of fire-resistant species. The second thing, again, we tested the growth of the 4 remnant plant species in these soil types, and we did expect differences in the growth of plants, partially mediated by these differences in the soil that we're likely going to see. And we expected plants to exhibit greater development in soils that had increased abundance of symbionts or a lack of pathogens. And our results here kind of can directly help managers understand and determine maybe the appropriate stage of reintroduction for these particular woodland remnant specialists. And lastly, again, we wanted to characterize changes in the soil microbial community based upon soil conditioning that the plants have done in the inocula. And so we expected the plants to develop very unique soil microbiomes when grown in these different inocula. And this, again, reintroductions alter the soil microbial community, which can lead to differences in long-term restoration success. So to do this, we had 4 experimental blocks of 20 square meters in each of those restoration sites, young, intermediate, and old. We collected soil cores and bulked them, and we assessed 14 different chemical and physical properties. We also quantified the basal area of all the trees in the canopy and the mid-story, and we identified all herbaceous woody species that were less than 1.5 meters tall. We classified trees by their known association with either AM or EM fungi. So again, trees can have associations with fungi which could drive differences in soil microbial communities. So we wanted to know what is the abundance and the density of these particular trees in these habitats. And we calculated the relative abundance of these trees, and again, we DNA metabarcoded the soil inocula. Then we had sterile pots in which we added some of the inocula to each one, and then we obtained seeds from the 4 species, Coreopsis pubescens, Geum virginianum, Solidego arguta, and Solidego caesia. We had 360 pots. It was a very large greenhouse experiment. And we used leaf area, which is a reliable proxy for plant growth, and it's non-destructive. And this part was really important because these pots were then placed in the field for a subsequent experiment. So we needed to not damage the plants as they were going out into the field. And then we DNA metabarcoded the soil from each species by inocula treatment to, again, look at the conditioned soil. The part I'm most excited about, which is probably the part that's most boring to all of you, is the methods for the DNA metabarcoding. So again, DNA metabarcoding allows for the simultaneous identification through the DNA of taxa within one sample. And so that can include anything from plants to fungi, bacteria, other eukaryota. But depending on what you're looking for, you use different primers. In this particular experiment, we were interested in the fungi and the bacteria. So we used ITS fungal primers and 16S bacterial primers. We assessed 2 separate meta-barcoding data sets, again, the inocula and the conditioned soil. I used CHIME to align all of these sequences of DNA and then extract reads and create ASVs, which are basically just little fragments of DNA that you have to then assign a taxonomic, with a taxonomic classifier. So all the fungal ASVs were assigned with the UNITE classifier, and all of the bacterial ASVs were assigned with the Silva classifier. So what did we find? Well, we found that young woodlands had a significantly greater density and basal area of AM associating trees, and this was largely driven by sugar maples, Acer saccharum. We also found that the intermediate and the restored sites had much higher concentrations of phosphorus and potassium than the young restored woodland did. But all other soil properties and all other vegetative properties were the same. There was no significant difference there. So really, relatively speaking, these sites are highly similar. But how do their soil microbial communities vary? Well, fungal diversity, first off, was significantly different. We saw significantly higher fungal diversity in the young restored site compared to the intermediate and old. However, bacterial diversity did not vary. So across all sites, there was no difference in the diversity of these taxa. However, this is pretty much standard. We see this in the literature quite often, where possibly the diversity of these taxa do not vary, but the composition of these taxa vary. So when we look at the Bray-Curtis dissimilarity of these populations, we see that for both bacteria and fungi, there is a significant different community composition of these microbes across these 3 habitats. And we can really dive into this with an analysis called the Simper analysis. So a Simper analysis allows us to determine which taxa or which taxonomic group is contributing significantly to those differences in Bray-Curtis dissimilarity. And so we can see here, when we look at the phylum of bacteria, that all of the Bray-Curtis dissimilarity contribution for proteobacteria largely was driven by differences in higher abundance of in young soil. So proteobacteria was significantly more abundant in young soil, and therefore it's driving those differences in community composition. We see something similar when we look at Firmicutes, which are heat-tolerant bacteria, and we see that it's largely driven by many more taxa occurring in the old, which is what we expected, right? They experience more fires. It's likely that these heat-tolerant bacteria are going to be more prevalent. When we look at the fungi, it's not as exciting. Unfortunately, there's a lot of things we don't know about fungi in terms of what taxa are there, or what taxa there are in the world. So there's a large portion of it that is unidentified. But when we look at these 2 main groups of saprotrophs, which also contain some pathogens, Basiomycota and Ascomycota, we see that there are large differences in the contribution of these taxa across the soil types. So then when we look at the forb development and how it varies when planted in these soil inocula, almost all the plants developed larger leaves when grown in young soil. So all except Solidagosacea grew larger leaves when grown in the young soil. And you can see that there is a slight increase of the plants, Solidagosacea, when grown in young, but it's not statistically significant. So this is really interesting in that we know the soils vary quite drastically, and we can see that the plants are responding to those. But then how are the plants then conditioning the soil microbial communities after they've been placed in these inocula? Well, the community composition of these plants in these soils are significantly different. So across the board for both bacteria and fungi, we see a response of a completely different community composition when we look at the soil-age interaction. And this part is really neat. So this was done with an indicator species analysis in which you can look at the number of unique core taxa that occur in one particular set or one particular treatment. And what we see here is that even the same plant species shows unique core taxa in a variable amount of numbers. So for example, Coreopsis pubescens planted in young soil has 35 taxa that are completely unique to it and no other species inocula combination. We see the same thing here, and even more so here. And this is occurring across the board, in which there are unique taxa that are contributing and associating with one particular species in one particular soil. And again, we use a simpler analysis, but in this example, we're using it to look at functional differences. So with fungi particularly, you can assign a particular function that it's known for in the soil. So we have pathogens here, saprotrophs, AMF, which we've discussed, and undefined, again, lots unknown about fungi, and EMF, ectomycorrhizal fungi. And what we're comparing here is the initial soil inocula and the conditioned soil. And what we see here, when we look at, we'll start with pathogens, there is a contribution from each soil type into the Bray-Curtis dissimilarity. However, it's exacerbated by old. And so what this means then is plants grown in old soil are cultivating pathogens, or potentially it means that plants grown in young and intermediate soil have an ability to not cultivate those pathogens. So we're seeing a shift here. When we look at AMF, any of the community composition differences that were driven by AMF were only occurring in young. However, when we do move to conditioned soil, we see that plants are able to cultivate AMF in some relatively high numbers, enough to contribute to differences in community composition in these other soil types. However, it is still largely driven by young soil. And when we look at EMF, ectomycorrhizal fungi, there's no contribution to Bray-Curtis dissimilarity. That doesn't necessarily mean that they're not existent. It just means they're not driving the patterns very strongly. But what we see here is when plants are placed in old soil, they're able to cultivate EMF enough that it's actually driving differences in those communities, which is really fascinating. And so kind of the takeaway here is that there's different abundances of these microbes across species. And so what we see here is with symbionts, certain species are cultivating different microbes in different ways. So Coreopsis pubescens had a really high abundance of Glomeromycota, which is the phyla associated with AMF fungi, as well as cyanobacteria, which is an important plant growth promoting bacteria, when it was grown in intermediate and old soil. But we see that these two plant species had a high abundance of this symbiont when grown in young soil. So their responses to soil are completely different. We see this again with heat-resistant bacteria. Solidago arguta had a really high abundance of these firmicutes in all the soil. They cultivate this particular phyla very easily. But GM virginianum only had a really high abundance of these firmicutes in intermediate soil. So again, this is really just highlighting these unique microbiomes that they're creating. And this phenomenon could not be even any more evident than when we compare the two Solidago species. So these two phylogenetically, morphologically similar plant species that are often used relatively interchangeably in restoration cultivated completely different communities when grown even in the same soil. So Solidago arguta had a really high abundance of these plant growth promoting bacteria in young soil, much more so than Assasia. And Assasia had a really high abundance of Glomeromycota fungi in old soil, while Solidago arguta did not. So we're seeing that these plants, which we typically would consider to be highly similar, are creating completely different microbiomes. And so, though there was really no difference in the microbial diversity, we see strong differences in the community composition of these woodland restored sites and their soils. And this is likely, again, influenced by the duration of restoration management practices, which shapes the above ground and the below ground ecosystems which we've laid out. And we see, again, a higher abundance of these fire-resistant bacteria in old woodlands, which could be outcompeting other important bacterial groups like plant growth promoters. We see a slightly similar pattern when we look at symbiotic fungi, but to a much lesser extent. And there's a higher abundance of these AMF fungi, which are symbiotic again, in young conditioned soil in general, which is likely leading to the better development of these plants that are grown in young soil. So plants may be more able to cultivate symbionts in this particular young soil, which could be important for selecting inoculum for restoration management because it could have important influences on plants that are slated to be reintroduced later. And plants are clearly cultivating their own soil microbial communities depending on both the host specificity and the inoculum. You can really think of it almost like a fingerprint in which the plant and the inocula kind of have this unique to their own microbiome, and the composition will vary even in very similar species. And plant species identity has been shown to be the best predictor of both bacterial and fungal community composition, though many other environmental factors do contribute to differences in community composition. Plant species has really been shown to be the #1 driver in a lot of systems. And even, again, morphologically, functionally, and phylogenetically similar species are cultivating completely different microbes. And this is really important as we think about management strategies as a whole and long-term management for restoration success. But much more information is needed to understand how these remnant specialists may respond to these distinct soil microbes, and then how they will do in restoration, which is great because, as I mentioned earlier, these plants were planted out at Shaw for the continuation of understanding how they will be successful or not in reintroduction. But overall, this kind of aids in our understanding of the relationship between management, soil microbes, reintroductions, soil microbes again, and on and off and on and off again, and how this is kind of a never-ending feedback loop that could at any time contribute to major shifts in these soil microbial communities. So there's many, many authors and people and collaborators to thank on this project, and I just had a really small part in it. But I'd like to thank Michael Saxton and the Ecological Restoration team at Shaw. I'd like to thank the UMSL Restoration Ecology class, which helped with all the plantings, the Bellwether Foundation for funding the meta-barcoding, the NSFRU and all the students who assisted with the greenhouse experiment. It was a lot of pots, a lot of time. Leighton Reed, of course, who is now at Virginia Tech, played a major role in getting this project off the ground. Lots of collaborators, of course, our funding sources, the Living Earth Collaborative, and always Shaw Nature Reserve. And with that, I'll take any questions.